Okay, so for today's lessons, we're going to look at this cell wall. So when talk about cell wall, basically means that it's not really an organelle. Cell wall is not really an organelle. It's a structure. Okay, it's a structural layer situated outside of the cell membrane. So sometimes cell wall we are claim it as the extracellular because when talk about cells means that the cell membrane. So anything outside the cell membrane actually not is not really a cells, but it's a structure outside the cell membrane. So extracellular things already. Okay, outside the cells. So where we can get a cell wall? Okay, so cell wall, you can see that cell wall present in most prokaryotic cell. Okay, so now the prokaryote that I name it here refer to the bacteria. Okay, refer to the bacteria. So in algae, plant, fungi. Okay, but rarely in other eukaryote, including animal. Again, animal we don't have cell wall. Okay, now when talk about types of the cell wall, type of the cell wall. Now for algae and plant, the cell wall is the cellulose cell wall. Okay, write down cellulose cell wall. Fungi is the chitin cell wall. Okay, bacteria use peptidoglycan. Okay, use peptidoglycan cell wall. So different. So for today, our focus on the cellulose cell wall. Okay, peptidoglycan cell wall later. Okay, you just need to know. Got this. We have this type of cell wall known as peptidoglycan cell wall. Full stop. How the structure, the structure look like. So we're going to learn it in an infectious disease when we deal with the bacteria. Okay, so we look at the plant cell wall. Plant cell wall actually consists of 3 layers. Okay, generally we call it the primary cell wall, followed by this secondary cell wall, and then middle lamella. Generally we have these 3 layers, but middle lamella will be shared by 2 plant cells. Okay. So if you look at the structure here, look at the structure. So you can see that we do have the primary cell wall. Can you see that? This part very thin. Normally primary cell wall will be thin. Can you see that? Thin cell wall. So those are the primary cell wall. So we have 3 cells. Cell number 1, cell number 2, cell number 3. So you can see that we have the primary cell wall. This is a primary cell wall. And then you can see that this part is the secondary cell wall. Could you see that? So it's a multiple layer. You can see a layer by layer deposition. It's a deposition of the chemical from this secondary cell wall. So between 2 cells, you can see that you have a darker zones here, which is a middle lamella. Middle lamella is something like the glue. Okay, it's a pectin that seal or glue two. Okay, glue the 2 cells together. Okay, can you see that? You can glue 2 cells together. So this part is known as the middle lamella. Okay, so what is the main components of the primary cell wall? So the main component of primary cell wall is actually cellulose. And you should know at this moment, okay, this is a complex carbohydrate make up of several thousand of beta glucose. So glucose, we have 2 forms of glucose. We have alpha glucose and beta glucose. Okay, so the one that form the cell wall is beta glucose. The one that we consume, the starch, is alpha glucose. Are you clear? Okay, for example, the glucose that we consume is alpha glucose, not the beta glucose. Okay, so beta glucose is a different kind of glucose that the plant use it to make the cellulose. Okay, and they are joined end to end by covalent bond. So this covalent bond is known as beta-1,4 glycosidic linkage. Now, at this moment, no need to know. But in the 3rd chapter that we are going to learn, biological molecule, then you have to know this, okay? So generally, they have these intramolecular hydrogen bonds between the adjacent cellulose molecule, cause them to adhere strongly to one another, overlapping parallel array, forming cellulose microfiber. And this cellulose microfiber in turn held together with a bundle of cellulose by hydrogen bonds again, it form the cellulose fiber. And this cellulose fiber actually will run in different direction, so increase the strength of the the cell wall. So we know that cell wall have high tensile strength. It prevents the bursting of the cell wall, correct or not? Okay, or the cells, the bursting of the cell because of this cell wall. Okay, so let's look at what actually means by this. Okay, let us show you. Okay, now, so for cell wall, cellulose cell wall, how it look like? Okay, first you have this beta glucose. So this beta glucose will join with beta glucose, join with beta glucose, join with beta glucose by the bonds known as beta 1,4 glycosidic bond. We're going to learn this one in details, okay, when we come to biological molecule. So this thing will continue. Are you clear? So after we form this, then the whole strand like this is known as one cellulose molecule. Okay, so we have 60 to 70 of this cellulose molecule. Okay, so this is another cellulose molecule. So they are relatively parallel to each other, and they have this bond that join them together. So this blue color bond is known as hydrogen bond. I will show you guys what's meant by hydrogen bonds next topic. Okay, so this blue color bond is known as the hydrogen bond. And because this hydrogen bond is between two molecules, right? So we have 60 to 70 of them, okay, linked together. So linked by what? Linked by molecular hydrogen bond. So what is inter? Inter means between. Between these 60 to 70 molecule, right? So where intermolecular hydrogen bond is the one that join them together. So this one we known as now linked together to form one cellulose microfibers. Are you clear? So means that this one is only one cellulose molecule. So we have 60 to 70 of them linked together to form one cellulose microfiber. And then this microfiber, cellulose microfiber, okay, linked together again, okay, to form, again, many of them, okay, many cellulose microfiber linked together by against the hydrogen bond. To form what? To form one cellulose fiber. Are you clear? Okay, so means that it's become bigger and bigger. Okay, you try to imagine first, you only have one. 60 to 70 of them join together to form one microfiber. And then this microfiber, can you see that this microfiber, a lot of microfiber join together, then it form one cellulose fiber. And you can see that this cellulose fiber later, how we arrange them? We arrange them, okay, in different direction. Are you clear? So this is one cellulose fiber. So that's why it creates the pore. Can you see that? You have the pore, right? This, right? Something like a pore or gap. So that's why we say that plant cell wall, they are fully permeable. And because of this property, because the arrangement, can you see that? In a different direction. Why is different direction will be better? Okay, different direction because when you press it, it won't actually cause the slippery amongst the molecule. Now, let me show you guys something. Okay, now what you can do now, very simple. Let me on my camera. Sorry, forgot. Now you can see me now. Let me get some things. Let's see. Now you can see that, okay, in front of me here. So this is one, okay, Why one pencil? The one pencil actually, you can see me right now. Okay, so one pencil here. So this one pencil represent one cellulose fiber. So if we arrange them in the same direction, can you see that a whole bundle like this? Can you see the whole bundle like this? So if I exert the pressure, they will slippery. Can you see that? They won't stay as their own shape. You try to imagine that you have, that means 10 pencils together and you press on it. Can you see that? It will be out of shape. Can you see that? But if I arrange them in the different direction, then when you press it, it won't be out of shape. Can you see that? Okay. You realize that? Can you see that? When I press on it, it won't be out of shape anymore. Because why? They have the resistance and also the friction. But if you put them, all of them at the same directions, can you see that? When I press on it, it will be out of the shape. Okay, can you see that? How I will put it? Okay, can you see that? It will be out of the shape. So this is because and why we need to increase. If we want to increase the tensile strength, then we have to make sure that they are running at different direction. And because of this, it creates the pore or the gap. So therefore, cellulose cell wall, we say that they are fully permeable. Now, we say fully permeable means what? Fully permeable doesn't mean that anything also can move in. Be careful. They must have a certain small molecule. Okay. So if you compare to the what we have learned in the cell membrane, so we do know that for the cell membrane, so cell membrane, they are selectively permeable. So why we say that? Because we have three categories of the items, right? Or particles that we have discussed previously. So selective permeable means that they allow non-polar molecule to diffuse across the phospholipid bilayer, non-polar CO2. Or we say that it's a lipid soluble particle. Okay, but they will block the polar hydrophilic particle, for example, glucose, amino acids, vitamin C, vitamin B, those are water soluble, they cannot. Okay, they also block what? They block the charged particle. Are you clear? The charged particles, for example, the ions, okay, you won't be able to cross or diffuse across the phospholipid bilayer. But how about cell wall? Cell wall, we say that they are fully permeable. So means that they allow non-polar particle, they allow the polar particle, they also allow the charged particle. But be careful, it's not like everything also can pass through. They have what we call the size exclusions limit. Okay, size exclusion limit means that they have a certain degree. Right? Let's say, for example, I have the pore, but doesn't mean that anything also can pass through the pore, like my door. So if let's say my door is this size, okay, then I want to elephant to pass through, difficult, cannot, right? So even though we use the fully permeable, be careful, we say that fully permeable to small, non-polar, polar, and charged particle. So they must be small. If they are large, sorry, cell wall also will exclude them. Are you clear? For example, okay, some chemical are too large to pass through. Okay, this cell wall, like very, very large proteins may not be able to pass through. Okay, so that's the idea of what's meant by fully permeable. Okay. So because of the arrangements, okay, so because of arrangements of these several layers of the fiber running in different directions, therefore they increase the strength, or we call tensile strength. Are you clear? So because if you put them together as same parallel, when you squeeze them or you exert the pressure to them, then definitely they will be out of the shapes. Okay, so the primary cell wall of most plant cells is permeable to the polar and non-polar, even the charged small molecule, including a very small protein, but they have the size exclusion estimated 30 to 60 kilodaltons. Okay, now what means the 30 to 60 kilodaltons? So you can imagine, okay, for water molecule, H2O, the molecular mass is 18 Dalton. Are you clear? Molecular mass is 18 Dalton. For glucose molecule, It's about 180 Dalton. So means that it can up to 60 kilodalton, means that majority of the small proteins, they will be able to pass through. But cell membrane cannot. Cell membranes, they exclude it and they are selectively permeable. Okay. And the secondary plant cell wall, which are often, deposited inside the primary cell wall as the cell matures. Okay, so the most common additional polymer in the secondary cell wall is called lignin. So particularly in the xylem. Okay, so the idea here is to make sure that the secondary cell wall can strengthen and also provides the waterproof to the wall, particularly in the xylem vessel elements. Okay, are you clear? Okay. So a specialized region associated with the cell wall of the plant, okay, so is we call as the middle lamella. Okay, middle lamella. So middle lamella, actually they are rich in pectin, shared by the neighboring cells and cements them firmly together. So basically we want them to join the cell together. Okay, so how about the plant animal cells? Animal cells, how they can join together? Because we do know that plant cells join together by middle lamella. So how about our animal cells? So animal cells use different way to join together. For example, we do know that we have the glycoprotein and glycolipids, right? So glycoprotein and glycolipid, actually they can interact together and join two cells together or stick two cells together. Are you clear? But because of the cell wall, then the glycolipid and glycoprotein are not exposed totally. Are you clear? Okay, so therefore, how they join together? They join together by this layer called middle lamella. Okay, so look at this, guys. Activity 1.29. So take 1 minute to label the diagrams. Okay. Okay, I assume that you guys have done this. Let us look at the answers. So for the first layer, gel-like structure. Okay, can you see that? Something like gel-like structure. And then this squeezy look like this, like agar. So this one is something like glue that we name it as the middle lamella. Okay, middle lamella. So the function is to glue or cement, to cement the two cells or two plant cells together. And then in this diagram, you can see that the cellulose fiber run in different directions. So this one we can call it as a cell wall or primary cell wall. Are you clear? It's not lignin, right? You didn't see a lignin, so it's either primary cell wall or the cell wall. And what's this structure? This structure. So this is the cellulose fiber. Okay, sorry guys, my mistake. This one is cellulose fiber, not microfiber. Okay, cellulose fiber. Okay, not microfiber. Okay, so then we continue to the structure here. So we have this special structure called Plasmodesmata, Plasmodesmata or singular Plasmodesma. So it's a microscopic channel that directly connect the cytoplasm of the neighboring plant cell to each another, establish living bridge between the cells. So this is very important, okay, because the Plasmodesmata have been shown to transport sugar, salt, amino acid, and some protein and messenger RNA from one cell to another cell. And it ensure or establish what we call the cytoplasmic continuity between the neighboring cells. And within this pore, those small molecules actually can pass through by diffusion. Okay. And majority of Plasmodesmata also contain a narrow tube-like structure known as the desmotubule, okay, which is derived from the smooth endoplasmic reticulum of the connected cells. Okay, so the desmotubule does not completely fill the Plasmodesmata, and it's very important because SER, something SER, right? So it involved in the lateral transfer of the lipid molecules from the SER from one cell to another. Okay, so the Plasmodesmata typically form during the cell divisions when the part of the endoplasmic reticulums of the plant cell get trapped in the new cell wall that is produced to secrete the to create the daughter cells. Okay, now let us look at a few things I want to clarify. One, what's the difference between the Plasmodesmata and also the pits? Okay, particularly when you look at the secondary cell wall and second, how the plasmodesmata can be formed, okay, during the cell divisions. And third, what is this desmotubule? Okay, that involve the lateral transfer of the lipids molecule. So look at the diagrams here. So you can see that we have different plant cells. Okay, we have 4 plant cells here. Okay, so if you look at the zoom into the particular region, you can see that this is the plant cell wall. We have middle lamella. Okay, we have the tubular structure known as the desmotubule. Can you see that? The desmotubule. So this desmotubule actually connect 2 smooth ER together, and we do know that smooth ER involved in the synthesis of the lipids, correct or not? Lipid synthesis. Okay, so how we transport the lipids between 2 smooth ER between 2 cells, then is through this desmotubule. Can you see that? So this desmotubule does not fill up the entire space. So it means that we still have the gap for other particle, for example, glucose, chemical to pass through, and for example, sucrose to pass through this gap or this pore. So this pore is known as the plasmodesma singular and plural plasmodesmata. So if you view it under the electron microscope, you can see the pore. Okay, so this pore is known as plasmodesmata. Okay, now because we haven't learned about the mitosis and cell division in plants, but generally when plant cells carry out mitosis, you can see that this is a plant cell. So they are elongated. So this is a cell wall. Okay, so after mitosis, they have 2 nuclei already, right? Mitosis, because it's a nuclear division, so they have 2 nuclides. So when they have 2 nuclides, what they need to do next is to form the partition between these 2 nuclei so that it can form 2 cells, right? So how they can form this? Actually, they build the cell wall through the vesicle. So the vesicle actually contains the cellulose, cellulose. So this cellulose we're going to join together. Then you can see that they start to have the vesicle, they form like this vesicle. So they line up the vesicle. So this vesicle contains the cellulose. So this cellulose later will join together and then start to form the cell wall. So after this vesicle, they can see that now vesicle or small vesicle join together to form a large vesicle. At the end, you can see that they form the cell wall with the middle lamella. So if we continue, it will be something like this. Okay. Can you see that? So perfectly become 2 cells now. Okay, the cellulose join together to form the cell wall. Okay, now this is a membrane. Okay, the blue color show that the plasma membrane when the cell is fully turgid. So they stick. Okay, they actually stick to the cell wall. So the blue color indicates the cell wall. Cell membrane. Sorry. Now, sometimes what will happen here is during the formations of the cell wall between adjacent cell, what will happen here is in between you have the smooth endoplasmic reticulum, because we do know that smooth endoplasmic reticulum, you can find them and they are actually located, dispersed, are all over the cytoplasm. So it's something like this. You try to imagine, okay, I built the wall. We are using the brick. I built the wall. But when I build the wall, what will happen here is, if suddenly I have 1 tube, okay, so means that I have to build the wall. Can you see that? I built the wall by actually forming this, the gap, the hole between, right? Because, allowing the tube to, okay, allowing the tube to be, so located there. So when this tube form, then you can see that my wall actually have a hole. Correct or not? Allowing the tube, okay, allowing the tube to be, okay, we don't cut, apart the tube, right? So this tube actually remain inside this. So the structure of the cell wall here, okay, is known as plasmodesmata. desma. And the tube remaining here is known as the desmotubule that connect to SER together. So this desmotubule allowed the particle to be shared between two cells. So it allowed the cytoplasmic continuity between two cells as well. You get the idea, guys? Do you get the idea? Okay, so it means that you try to imagine that you try to stick, I mean, to build the wall, but suddenly you got one pipe there. Okay, and you cannot, I mean, remove the pipe, right? If you cannot remove the pipe, then you build the wall, but you're going to, if you continue to build the wall, can you see that? Okay, continue to build the wall. Okay, so but you create the gap now. Can you see that? You create the pore in this case to allow the tube to pass through. So this tube is known as desmotubule. Okay, so means that at the end you can see that it may create the pore like this. Okay, and then cell membrane will continue. Can I see that? And you have the SER, for example. Okay, SER, the tube. Okay, they are interconnected. Sorry, SER, tube. Okay, and then continue this tube to another cell. Okay, tube. Okay, so this structure is known as plasmodesmata. So plasmodesmata is an open pore. Okay, so particle can move in and out, okay, from one cell through this plasmodesmata. So we do have another structure known as pits. Students always confuse between these two, the pits versus plasmodesmata. So what's the difference between the pits and plasmodesmata? Pit is not an open pore, but plasmodesmata is an open pore. So if I draw the structure, what's pit? Pit basically is the regions of the cell wall without lignification. Okay, are you clear? Okay, so now we look at the structure in terms of the pits. So because we need to identify it, right? So we do know that this is a plant cell, okay? Transverse sectioning of the plant cell. So plant cells have the cell wall. As usual, you need to draw the cell wall, a very thin primary cell wall. Okay, this one cell, and then we have another cell. Okay, plant cell wall, very thin primary cell wall. Okay, so I labeled it as the primary cell wall. Okay, now how about the secondary cell wall? So secondary cell wall, for example, lignin. So lignin will deposit. So when the lignin deposit, it may not be complete. Okay, lignin's deposit. So layer by layer, it deposit already, but sometimes some parts that without the lignin. So form like something like this, okay, without the lignin. Okay, so if you label, this one is the secondary cell wall. Can I see that? Depositions of lignin formed by lignin. First primary cell walls formed by cellulose. Are you clear? Okay, lignin is waterproof, so water cannot pass through now. Okay, so that's why normally the cell will die. Okay, now this is second cell. So second cell also have the lignification. Okay, so the regions that without the lignifications, this region, can you see that? This region, this region, this region, those green color or arrow indicate that this is the pits. Are you clear? Can you see that? Is it open pore? No, it's not open pore because you still have the primary cell wall there. It's not an open pore. Are you clear? So this structure is known as the pits. So be careful when you label the diagram. So you check whether there is primary cell wall or not. If there is a primary cell wall, then it's not open pore. when it's not open pore, we cannot claim it as the Plasmodesmata. So how the Plasmodesmata actually look like? You want to take a diagram? I take the photo. You can take these pits first. Okay, students always confused about this structure. Okay, now we look at Plasmodesmata. How the Plasmodesmata look like? Plasmodesmata, they are open pore. So means that I have a cell, first cell, I have the second cell. So they have cell wall, primary cell wall. Normally, the cell with the Plasmodesmata, they don't have the lignification. So they only have the primary cell wall without secondary cell wall. So made by this cellulose. Okay, formed by cellulose. And because we know that when formation of the cell plates, right, the cell wall is incomplete because of the desmotubule, we call the SER. So they have the pore in between of them. So this pore, they are open pore. Can you see that? So the arrow here indicates these are the Plasmodesmata. It's an open pore. Okay, okay, clear? So I zoom, make it smaller. Then you can check. Okay, you can take a photo for these two. Okay, the differences between the pits and the Plasmodesmata. So Plasmodesmata, they are open pore. Right? They are open pore, so they can actually allow the continuity, okay, of the cytoplasm between two plant cells. Are you clear? Okay. Any questions so far, guys? So no questions, then I off for the first sessions. Okay,